would you like to go for your walk? Hello everyone, thanks for coming back to watch this video. Today's video is going to be all about genetics and how genetics is involved in Fragile X. So I will not lie, today's video is going to be dense, but we'll try to make it succinct. So if genetics sounds very intimidating to you, please stick with me because we will walk through it step by step. And I am currently in the lab and have to wear my lab coat, and I'm doing honeybee research, so this is why it says Isabella and a honeybee, and why I'm wearing my glasses. With Fragile X, remember there's a funky end on the end of the X chromosome. This is what a normal X chromosome looks like, and this is what a fragile X chromosome looks like. You can see there's this little funky, weird, like tail at the end on both chromatids. So this is where the FMR1 gene is located. And remember that Fragile X is associated with mental retardation in the full mutation, and mental retardation is linked to the FMR1 gene. So now we're looking here at that tail. That's the location of the FMR1 gene, and that is where those excess CGG repeats are located as well. Because of the excess CGG repeats right here, this causes not enough FMRP proteins to be made, and this causes a problem. Now let's unpack that because that is a lot, that's a mouthful of words. In order to do that, we're going to need to do a little bit of a genetics review. So what I have done, remember that chromosomes 1 through 22 are autosomal chromosomes. Those aren't sex chromosomes, meaning they're not an X and a Y. The 23rd pair of chromosomes in human DNA are the sex chromosomes, and that's what you see right here. A male has an X and a Y chromosome, and a female has an X and an X chromosome. Now let's say this male and female have a son and they have a daughter. The son, this is how it works with everybody, by the way, this is not just this special family. So the son gets his X from his mother and his Y from his father. It has to be that way. Same thing over here, the daughter gets her X from her mother, and she also gets the father's X, because if he gave her his Y instead of an X, she'd be a boy, right here. Remember transcription and translation from high school biology? Maybe, maybe not. But what's important to remember is the central dogma of biology. The central dogma of biology is all about how proteins are made. Step 1, DNA is used to make RNA. In step 2, RNA is used to make proteins. So RNA is the intermediate between DNA and proteins. Our body is full of proteins, functions on proteins. They are very, very important and fundamental in order to carry out daily functions. In this case, we're talking about DNA in order to make FMRP proteins. So DNA in the FMR1 gene encodes for RNA to make FMRP proteins. We also have to recall the nucleotide base pairing system. So we have 4 bases. We're going back to high school biology. A, adenine. Which way is my hand going? T, thymine. C, cytosine. And G, guanine. So A always bonds with T. T always bonds with A. C always bonds with G. And G always bonds with C. And when I say bonds, the bonds that are forming are called hydrogen bonds. And those are what are kind of like the rungs in the ladder on that classic DNA model that we all know so well. These are the rungs of the ladder, those are the hydrogen bonds. With Fragile X, what we are concerned about is a trinucleotide sequence that is CGG. So that is why I've been saying CGG repeat, this is it. So CGG, of course, bonds with GCC. They're not drawn, but there should be little hydrogen bonds right there, and they would be flipped and on this ladder too. Now on the FMR1 gene, 55 to 200 CGG repeats means that an individual has the premutation. I'm 100, that falls within 55 to 200 repeats. If an individual has 200 or greater than 200 repeats, then that puts the individual in the full mutation range. Your CGG repeat is really important for your doctor to know exactly what that number is. It's really important because it determines a few things. Number one, of course, it will determine if you are a premutation or a full mutation person. Number two, it will determine your likelihood of getting FAX-POI, like I have, Fragile X primary ovarian insufficiency. And it also determines your likelihood of getting FAX-TAS, which is Fragile X associated tremor ataxia syndrome. That appears as Parkinson's, but it is not Parkinson's. So it's important to know that it's not Parkinson's because it's different, it's treated much differently. And finally, one of the most important things that it can tell you, your CGG repeat number that is, it can tell you the likelihood that if you pass on your Fragile X, what is the likelihood that your CGG sequence on that Fragile X will expand to a full mutation, meaning your child will be full mutation for Fragile X. So how is Fragile X inherited? Its mode of inheritance is X-linked dominant, hence the name Fragile X. It depends on whether the father or the mother has the premutation of Fragile X and will likely pass it to the child. The father has the premutation because Fragile X is X-linked. A father cannot pass his Fragile X premutation to a son. Because think about it, a father has an X and a Y, and Fragile X is carried on the X chromosome. So if he has a son, the son will have his X from his mother. He'll inherit his X from his mother, not his father. And his father will only be giving him his Y. So if Fragile X was carried on the Y, if it was Fragile Y, then the son would be able to inherit it, but that is not how it works. So a son can never inherit the premutation from his father. However, a father passes his only X chromosome to a girl. So all females will have Fragile X, the premutation. When a father with the Fragile X premutation passes his Fragile X to a daughter, the CGG repeat sequence will either stay the exact same, the same number, or it will contract. It can't get bigger. So when it contracts, say the father has a CGG repeat of 100, then he has a daughter, that CGG repeat can either be 100 exactly or less. For me, my father had a repeat of 100, and so do I. So I'm just an example of that repeat sequence staying the same and not shrinking or contracting, as I said. The mother has the premutation. There is a 50-50% chance that the child will inherit the Fragile X, not the normal X. So it doesn't matter whether you are pregnant, if you're the female and you have Fragile X, if you're pregnant with a boy or if you're pregnant with a girl. If they inherit your Fragile X, it doesn't matter what their gender is, they're still inheriting an X from you. So that's a toss-up of whether they're going to inherit your normal X, because you do have a normal X, that's always a possibility, or they could inherit your Fragile X. 50-50 toss-up, kind of like a flip of a coin, really. With a mother having the premutation, and this is how it will work with me, the CGG repeat will always expand. But the question is, will it expand just a little bit, or will it expand so much that it is considered a full mutation? I really, really hope that my explanation was a good one and that it helped to maybe clear up any confusion that people might have about genetics, and I hope that it put somebody's mind at ease trying to learn about how Fragile X is inherited. You do have a 50-50 chance of passing your normal X, so that's something that I didn't realize originally, and that's something that my genetics teacher sat down with me and helped me understand, and that completely changed my mindset, is there is a 50% chance that you pass your normal X. So it's a toss-up. Anyway, thanks for watching, and I'll see you again soon. Bye-bye. and chromosome pair 23 are